In terms of like my science journey, I guess, I'm from Durham originally, and I just really liked my science classes. I wish I could give you like, this is the thing that made me really interested in science, but I just think it's cool. And so I decided to like keep going with that. I sent my resume to Duke Biology, which sounds impressive until you realize that a high school resume has like your standard high school classes on it. But because I did that, I was able to get a position in a lab at Duke. So I started in Shin Dong's lab, and she works with plant microbe interactions. So I did that for a couple years in high school and then continued over the summer and like winter breaks during undergrad. I started undergrad at Davidson College in 2011. Davidson's in North Carolina, right outside of Charlotte, and it's about 2,000 students total. So again, if you haven't heard of it, like Nick said, I'll stick with the basketball theme. We're known for Steph Curry. Small shout out there. So I did my undergrad at Davidson, and then I started grad school at Duke in 2015. I'm a 6th year PhD student, and I'll be starting a postdoc position also at Duke in September. So I'm thinking of sticking with academia. So that's science, but there's really a lot more than science, and I won't go through all of this. I've done tons of stuff. If anybody ever wants to talk about time management, I'm your person. Some things during grad school, I guess, that I'll highlight. I've been really involved with the graduate and professional student government community pantry. So something that a lot of people aren't aware of is that there are undergrads and grad students that are food insecure across the US at various institutions. And so we've done a lot of work trying to figure out what that number is at Duke and what are some solutions to help address that. I've also been really involved with outreach. I was the outreach director for my department and also for the graduate and professional student government for a while. And then fun fact, so I majored in biology, but I also have a minor in dance. It's not all science. And so our lab is really interested in how bacteria are interacting with their environment. In most cases, we actually look at bacteria in a human environment, so pathogens. But for this project, we've actually changed our focus a little bit to look at the environment and like specifically interactions with plants. And so something that's really important to keep in mind is that in each of these different environments, bacteria are going to face a lot of different types of stress. So like in the clouds, for example, it might be really cold, but in the gut, you might be facing acidic conditions. And so bacteria have to adapt to these in order to survive. A major barrier against all these different types of stress is the outer membrane. And so for gram-negative bacteria, they have an inner membrane and an outer membrane separated by a thin layer of peptidoglycan. And this is like the first barrier against all these different types of stress, whether that's pH or whether there's a really high salt content around them, whether it's really hot, things like that. And even though we normally think of or depict these membranes as just a single line that surrounds your like bacteria, it's actually a really complex environment. And so in the inner and outer membrane, you have phospholipids. The outer membrane specifically has lipopolysaccharide or LPS. And the LPS really helps create a strong barrier against these different types of stress. There's also a variety of proteins and different lipid species in the membrane, and those all have implications for how that membrane is able to function and protect the bacterial cell. And importantly, bacteria can modify this membrane in order to respond better to the environment they find themselves in. We want to know how bacteria interact with their environment, but we're really interested in these extracellular vesicles as a way to interact with the environment. And so these vesicles for gram-negative bacteria are termed outer membrane vesicles, as Nick was saying, and they bud off of just that outer membrane. So in general, they don't include inner membrane, although in biology, I don't think you can ever say like, this never happens. So you can think of them as only containing outer membrane. And typically they contain any of those proteins or lipids that are found in the outer membrane. They can serve some purposes for the bacterial cell, like relieving membrane stress. So in this picture, I'm showing a buildup of misfolded proteins in the membrane, and then those get packaged in the vesicle as a way to relieve some membrane stress. Bacteria also package specific proteins in those vesicles that can be used for virulence to help attack a host, attack a plant cell, attack a human cell, and that can help the bacteria survive. They serve a variety of purposes though, and that's what's depicted up in that top left kind of. And so we know that they contain nucleic acids, and this can lead to horizontal gene transfer. They can serve as decoys against bacteriophage or antibiotics. So if your vesicle absorbs all of the antibiotic in the area, then the bacterial cell is free to go about its normal life. And then the one that I'm most interested in and my project focuses on is their ability to package toxins and other molecular cargo. In particular, I'm interested in toxins that might elicit an immune response in plants. So that's kind of the broad picture. How do bacteria interact with their environment, and how do vesicles play a role in this?